Good morning, everyone. Today, oh my. Good morning, everyone. My name is Juliet Morrison, and I'm an assistant professor at University of California Riverside. And today I'll be speaking to you about how lung immune populations can drive influenza outcomes. I will also demonstrate how one can actually use gene expression data, bioinformatic tools, and statistical analyses to generate novel hypotheses about immune cell involvement in viral diseases. So for a little background, viral disease severity is determined not only by the virus, but also the host response to that virus. So it's a very delicate balancing act, which means, therefore, that too much of a response is bad and too little of a response is bad. You have to have a delicate balance. One of the goals of my research is developing host-targeted viral therapeutics. So what I mean by this is that if you can imagine that viruses versus hosts are like children on a seesaw, in this scenario here, we have the host response being stronger than the viruses. But at any moment, we could have additional viruses coming in and overwhelming that host response. If there's too much of a host response, however, you will also get tissue damage and disease. Today, I'll be talking about seasonal influenza. It results in 3 to 5 million cases of severe disease and 250,000 to 5,000 deaths annually. And it causes a great economic burden. About 11.2 billion in economic costs per year in the US are related to influenza infections. So I was interested in finding out if there could be any signatures that could distinguish influenza severity based on their pathogenicity and serotype across a wide variety of influenza viruses. And so to do this, we combined two different studies of very similar design, where in one study, we have mice being infected with different influenza viruses, some avian and one human. And in the second study, we have a variety of H1N1 human viruses. And then we take these data, we take this, and then we harvest the lungs from these mice, and we look at the viral titers, and we look at histopathology, and we do transcriptomics. And additionally, we are also taking weight loss and survival data. Once we combine these data, we then integrate everything to come up with a larger experimental. We then combine these data to come up with a larger experimental data set. And I want to point out in the left-hand corner, I'm showing you a weight loss curve of the different viruses in this particular compendium. We have a variety of viruses that lead to severe weight loss and death in mice. So the sad face with the crossed out eyes. We have viruses that can lead to some weight loss, but then the animals recover. And then we have other viruses that cause very mild disease. And so the mice do not lose weight in this situation. And so we did the transcriptomics on the lungs of these different mice, and we found that we found that there were increases in the inflammatory response in the animals that had more severe disease. We also found, quite strikingly, that there was this decrease in coagulation and lipid metabolism gene expression in the animals that developed severe disease. And instead, we saw an upregulation of certain lipid metabolism and coagulation genes in the mice that developed mild disease and survived. And this we found, we have highlighted here in the black box. So what does this even mean? Well, why are we seeing these transcriptional differences? Well, the first thing to consider is that the lung is an amalgam of different cell types. So we have multiple epithelial cells, we have different immune cell populations. And we wanted to find out which of these populations was causing these differences in transcriptional responses that we were seeing. And to interrogate this, we used a tool called Digital Cell Quantifier, or DCQ. And what DCQ does is that it takes an immune cell compendium from ImGen with many different immune cell types, and then it combines it with your own genome-wide gene expression data, so our lung transcriptional data, and then they infer the in vivo global dynamic changes in the immune cell quantities within your lung tissue. So basically, DCQ can predict the temporal process of an immune cell response to stimuli within a mouse model. After we got the results of the DCQ, we then used statistical analyses to correlate the presence of these different populations from ImGen with the weight loss that we found in these animals. And you can see here that we see that a prediction of lung DCs being decreased as the disease intensity increases. We also see that DCQ predicts that granulocytes will increase in the lung in more severe disease. And we also see that alveolar macrophages are predicted to decrease in severe disease, and monocytes are predicted to increase in severe disease. And these are things that we already know. And I'm showing you here how we validated some of these results. So we take our mice and we infect them with either Cal-09, which is a mild flu, or with PR8 to give a severe disease, or we use PBS. And then we harvest the lungs at day 3 and we look at the different immune populations that were predicted by DCQ. We found that our data matched what DCQ predicted, in that we found that CD11C positive Siglec F positive cells, aka alveolar macrophages, were highly abundant in PBS infected or PBS controlled mice, were decreased in our California 09 infected mice, and severely decreased in our PR8 infected mice. We also found that the granulocytes were lower in quantity in PBS treated mice and were much higher in our PR8 infected mice, as has been shown previously. And finally, we also showed that the Ly6C positive monocytes are greatly enriched in the lungs of our PR8 infected mice. So with more severity, we see more monocytes. Thus, alveolar macrophages, monocytes, and other immune cell types correlate with the disease severity we're discussing here. Now, I also want to bring your attention to this population here in green. This is the small peritoneal macrophage population. So what would peritoneal macrophages even be doing in the lung? Well, it turns out that the peritoneal cavity, which surrounds our abdominal organs, is closely related to two other serosal cavities, the epicardium as well as the pleura. So the pleural cavity is the serosal cavity that surrounds our lungs. And in fact, the cells of the pleural and peritoneal cavities are quite similar. Both the pleural cavity and the peritoneal cavity contain macrophages, such as the small macrophages and the large macrophages, and they're distinguished by their size as well as the markers upon their surfaces. So how do we sort pleural macrophages? Well, in my group, we take live cells, single cells, and then we sort for cells that are CD115 positive and CD11B positive. And then within that population, we separate them based on MHC class 2 positivity or F480 positivity. So small pleural macrophages are MHC class 2 positive and F480 low, while the large pleural macrophages are F480 high and MHC class 2 low. Now for some real experiments. So we wanted to ask whether this population that resembles our peritoneal macrophages are actually pleural macrophages. And so to do this, we took a very simple approach. It's actually very hard to target the pleural cavity, but we have actually developed a procedure in the Morrison Lab where we can introduce dyes and other things into the pleural cavity without damaging the lung. So in this experiment, we are doing an intrapleural injection of a macrophage dye called PKH. And when you harvest the pleural fluid, you can see that we have these red cells here because the macrophages have specifically taken up this dye. Now, if we do that same injection of the pleural cavity with our PKH and then follow up with an intranasal infection with our with mild or severe flu strains or PBS, we find that after we remove the lungs and then look at them under the microscope, we find that we don't see any red cells in our PBS treated control mice, but we see a great deal of red cells, aka the pleural macrophages, in the lungs of our California 09 infected mice. And there are much lower numbers of these macrophages in our PR8 infected mice. So these are the mice that get severe flu and die. And this actually supports our initial prediction, because we had predicted that in the case of severe flu, that we wouldn't be seeing this pleural macrophage population. And we further followed up by looking at the histopathology of these mice and found that in the case of our California 09 versus our PR8, we see a lot more debris and bronchiolar collapse in our PR8 mice, showing that this is indeed severe flu versus mild flu. So what is the phenotype of these trafficked macrophages? Well, we went ahead and did flow cytometry on these populations, and we found that our PKH positive macrophages were actually looked a lot like alveolar macrophages. So Siglec F positive and CD11B low. And we also found strikingly that the macrophages that had accumulated in the lung were also MERTK negative, as opposed to the general MERTK positive macrophages. We're not quite sure what that means yet, but it's an interesting observation. And now if we take the flow cytometry data and we graph it, you can see more strikingly that in the pleural cavity with the addition of our viruses, we see a decrease in the numbers of pleural macrophages. And in the lung, we see this increase of the pleural macrophage populations, and especially with our California 09. So we see an accumulation of macrophages that originally originated from the pleural cavity, and they have now accumulated in the lung in the case of mild flu, but they do not accumulate in the case of severe flu. We then asked if pleural macrophages could have an impact on influenza outcomes. Does the fact that they move from the pleural cavity into the lung actually impact our outcomes? So to do this, we injected the pleural cavity of mice with either clodronate liposomes or with PBS liposomes. And these clodronate liposomes, when taken up by macrophages, will induce apoptosis in the macrophages. So we're basically clearing out the pleural macrophages from the pleural cavity. And when we do this, we actually see very targeted macrophage depletion. Because if you look at the plural macrophage population, both the small plural macrophages and the large plural macrophages are decreased in the pleural cavity. But we don't see any impact on our lung macrophage populations or the populations of the draining lymph nodes. So pleural targeting with clodronate is very, very specific for pleural macrophages. So with that in hand, we went ahead and we did an experiment where we do intrapleural injections of the clodronate liposomes versus our control liposomes. And then a day later, we infect with our mild flu strain, the California 09. And then we weighed the mice over the course of 5 days. And we found that in the case of the mice that had their pleural macrophages depleted, they lost weight more quickly than the mice that had the control liposomes, implying that the pleural macrophages do serve a protective function in influenza. We also looked at the viral titers in the lung tissue, and we found them to be pretty comparable for both the clodronate and the PBSL treated mice, suggesting that it's not necessarily a difference in the viral titer that's driving this, and most definitely a host response. And finally, I'd like to summarize what I've discussed today, which is that the pleural cavity may serve as a reservoir for lung macrophages. And in this case, we found that both small pleural macrophages and large pleural macrophages appear to cross the mesothelium and enter the lung, which is a very new idea. Furthermore, we know that they are having an impact on influenza resolution and influenza outcomes. How this is happening, we don't know. It has actually opened up a much larger, many more questions, because we don't know what's driving the macrophages into the lung, and we don't know what kind of cells they're really becoming in the lung. And we also are trying to figure out the mechanisms of how it's actually alleviating the severe disease. And finally, I'd like to acknowledge the members of my group and our collaborators who have been involved in this project. Thank you so much for listening, and have a good day.